Welcome to our first cherry nutrition webinar for July. I'm really excited to be delivering these as part of the extension project with the help of Cherry Growers Australia, Gita, and the rest of the project team. But I thought this would be an excellent way to kind of get us all connected again, looking at a topic that will be relevant for the upcoming cherry season. I cannot believe that we are literally only weeks away from flowering. So now's the time to get into your office, start planning what you're going to be doing for the season coming up. And we hope that some of these nutrition webinars gives you a little bit of either a refresh on things that maybe you've forgotten about, but you know are in the back of your mind, or something new that you might like to try for the next coming season. We're trying to make these webinars as engaging as possible. Sometimes it is difficult when we're online to be able to chat, but I encourage everyone, if you've got any questions, pop them in the chat. If you feel bold enough when it's question time, come online, to unmute yourself and ask a question. But I've got two brilliant speakers today to set up the series for us. So I've got Andrew Smith and Zen Kynigos, both talking about managing tree physiology for optimal nutrient uptake. And I'm going to pass over to Andrew first, and I'm going to stop sharing my screen. And so Andrew, most of you probably know Andrew. He's a senior agronomist with Hybrid Ag. And tonight he'll be specifically talking about knowing your cherry physiology for nutrient uptake. So thanks, Andrew. I might get you to share your screen, and it looks like you're
All
good to
right.
go.
Good evening, everyone, and welcome to the first of the series. So great to be a part of, and let's hope everyone gets out of the series some really good information. So, which I'm sure they will. So, anyway, we'll get on with it because I've got a bit to get through. So, cherries are a pretty big topic when you start to look at their growth stages from start to finish. So, um, we'll move right into it. So I guess the goal tonight is really to get everybody to understand the cherry physiological growth stages throughout the season a little bit better, just so you can understand sort of things like the right nutrition for the right stage. So that's, I guess, the aim that I'm trying to get across tonight. So then we sort of look at our bit of our learning map for where we're going to go to tonight. So we're going to look at the cherry phenological stages. So we're going to look break them down into dormancy. We'll just have a quick look at some soil considerations, reproductive phase, growth phase, and then we'll look at our post-harvest aging phase. So as we move into our cherry phenological stages, as you can see here on the screen, when we break it down, we've got our quiescence phase, which is currently known as dormancy. Then we move into reproductive, which is bud and flower, fruit set and cell division. Then we look at our growth stage, which is made up of cell expansion, fruit fill ripening, bud initiation. Then once we get past the bud initiation and harvest, we're actually looking into that post-harvest aging phase, which is where we can actually rebalance and move into senescence as well. So to try and put it all together and understand how this happens really quick, I've got a little bit of a table here, which I've put together, and we've got some bloom dates if we look at down the left-hand column there, and depending on whether they're low chill or sort of medium to high chill varieties to where they sit for bloom timing. But then when we look into, we say, our cell division stage there, we've really got a 20-day window from days after full bloom in our lower chill type varieties. And I've got Royal Toga listed as one there. When we move into things like Samba and Bing, we're somewhere around that 25-day, and then sort of Lapin and Regina 30 days. So we've got a window of 30 days to maximise that reproductive cell division phase there. And then when we move into that growth stage, for all varieties, it's sort of somewhere around that 40 to 55 days. And, that's what it takes to do that cell expansion phase and get the fruit basically to harvest. So when you look at some of these varieties from full bloom to actual harvest, we're looking at 60, 65 days or 80 to 85 days, depending on the actual variety and whether it's sort of a low, medium or high chill variety. So it really does happen very quick. So when we think about that, it really is it's like the cherry seasonal race. So here we've got to get down the track as quick as we can. We've got lots of things going on around us, and we need to be able to make sure we pull the right levers at the right time. So it is like a bit of a race. So we move into the quiescence or dormancy phase. So although there's not much action in dormancy, we don't have zero sap flow. We do still have a little bit, although it'll be minimal. And this is where the tree is obviously taking on its chill requirements, which after today, I'm sure there would have been a few more chill portions going because it was a chilly day here in Wangaratta. But it's very important that we've got right nutrient reserves sitting in the plant in dormancy, ready to go for when we actually get into that other next phase, which is basically where we move into reproduction. So just before we move into reproduction, I just want to really quickly touch on soil considerations. And once again, it's a bit like the race team. You know, we've got a race team which has got all the hardware in the pit crew. Well, we've got to make sure that our soil has got the hardware in it, being our nutritional type profile in the soil, and the pit crew within the soil, well, they're the biology. So we've got to make sure that we've got all the appropriate nutrients and the biology set there, ready to work in harmonious synchronization there to help deliver those nutrients into the tree once we actually get into that major growth stage. So super important that we're balanced up, ready to go. We've done a soil test. We understand what our limitations of our actual soil potentially is. So it's a really good point to start, particularly through that dormancy phase. So when we move into the reproductive phase, it's something that we really need to consider, that it is quite different than growth, and it has a different hormone expression than what our growth phase is. And we'll talk about that as we get a bit further down the track. But so reproductive really is about that bud development, pollen production, and then fruit set and cell division. So it's super important that we understand how that mechanism works and what sort of things we need to have in place and why we would apply something over something else in that space. So to try and understand that a bit more, we look at sort of reproduction versus growth in our tree, in our cherry tree. We'll have some synergistic relationships with nutrients based around cytokinin and or auxins. So it's really a balancing act. So in that reproductive phase, we want to try and have more of a cytokinin shift within the tree. So elements like manganese, phosphorus, ammonium type nitrogen, and sulfurs are what we would class as more reproductive type nutrients. And then we look on the growth side of the fence, which has got more of a synergistic relationship with the auxin production. We've got things like nitrate, nitrogen, chlorides, and things like potassium, which are more all coupled around growth elements. Whereas calcium, he's a guy that sits in the middle, so he can sort of balance one way or the other, and he's important for both sides of the fence, but can also be a reproductive or a growth element. So it's important that we understand when we are applying nutrients that we are applying them in the right form to maximise that reproductive phase. So if we move into this spring activation area, which is where we see rising temperature and longer daylight hours, we start to see some minimal slat flow come, and we can see bud development. So you can see on the page there, or the slide there, we start to see bud break, and we move into that early flowering. So this is really that process which is internally driven by the nutrient reserves which are being stored in the tree on the other side of the post-harvest side of the season into senescence. It's like that storage, just like a bit like a battery pack. So this is where the plant is really drawing out of reserves. We're not photosynthesizing at this stage, so we're not actually able to really draw up. We don't have transpiration in the tree at this stage. We don't have leaves. So as part of the reproductive phase, it's very important that we understand that we need to maximise our pollen production and make sure we've got really good, strong, viable pollen. And it's super important that we've got things like calcium that are really available to help drive that pollen production and other key considerations like water, good water supply, and obviously our pollinators, which are our bees, and then obviously the other things which really can, I guess, detract from that pollen and fruit set is things like frost or heat or humidity, which is stress event type things which come into the tree, which I know Zen will talk about probably a bit later on. So once we keep moving through this reproductive phase, it's a really critical part in the transition of the tree in that reproductive cycle. And once we get to that sort of full bloom phase, we've actually got over 50% of our fruit cells already formed through that full bloom fruit set phase. So once again, it's an area that we need to make sure that we've got all the goalposts aligned, because this is an area that we can maximise outcomes when we're not really typically think of what's going on. So it is important that those storage reserves are really well placed to drive this process through that flower reproductive phase right through into shutfall, and we're just starting to get some of that early leaf emergence. So, failures in this space really got potential to limit yield, no matter how well the tree grows later on in the growth stage. So then we look at the cell division phase, which is really engine room of fruit set and size. So it really sort of occurs that 14 to 21 days post up to post full bloom. Super important. So we really can support that cell division phase by making sure we've once again got things like calcium in place. It's very important that we're trying to maximise that calcium input through this time early in the season, because it really is foundational nutrient to make sure it's there for cell wall strength and so forth like that. So you, once again, as a grower, you really got that ability to influence this reproductive phase to maximise that cell division. And as we know, cherries don't get any more cells once they get past this cell division. We only really start to expand them. So if we can maximise this cell division period, it gives us the ability to not only grow bigger cherries, we grow firmer, harder cherries with more density in them as well once we get to a harvest, which on the other end helps our shelf life as well. All right, so we'll move to this next slide, which is, I guess, it really does explain that genetic difference, I guess, between larger fruit and smaller fruit. That genetic difference is really based around cell numbers or cell count. So we really, like I touched on earlier, we really want to try and maximise the reproductive of the cells and make sure we can get as many cells into the fruit as we can. Because then when we do actually get to the next stage and we start expanding cells, we actually have a firmer, solid piece of fruit which we can take to market. And, as growers, we actually can influence this space, so it really does need to be considered. So we look at really quickly what nutrients are pretty key around this fruit set cell division phase. We've got things like calcium, phosphorus, boron, manganese, zinc, cobalt, copper, and also things like fish kelp and fulvics. Although they're not actually nutrient, they do provide certain things that are really plant friendly. So once again, they're really key to set yourself up in that phase. All right, so we'll keep moving. We'll move straight into the growth phase, which is where we come out of reproductive. So we've got our cells all in our plants, or in our fruit, I should say. And now we want to move into this phenological stage, which we class as growth, which is about cell expansion. We move into fruit fill and ripening. And then also around that back end of early harvest, late harvest, we'll be actually thinking about bud initiation as well. So, there's a lot going on in this period. Really makes it or breaks your crop finish and the start for next year's yield potential. So when we move into this phase, it's really about photosynthesis. So you need to think of our tree as a solar farm. We really are farming for light, which is really managing that photosynthesis, which is plant energy or actually fueling the system. So, no different than the race car going down the track that's got to pull in and get fuel. We need to make sure that the tree is actually fueling itself to to drive the system. So if we have a bit of a quick look at photosynthesis, I'll explain it really quick. Obviously, we've got sunlight and carbon dioxide, which we want to take in through the leaves, and we want to bring water in through our roots. And on the other side of the slide there, we can see we're manufacturing sugars, which go into the tree and also back into the roots as exudates, and we also give off oxygen. It's a pretty interesting process with the leaf does, and it all comes from the leaf blade effectively, which is our solar panel. So we want to make sure that we've got great leaves that can actually create that solar efficiency from our light, which we're trying to turn into energy. So on our leaf blade, we've actually got little stomata, which effectively is it's like a valve which opens and shuts on the leaf, which will and it controls that gas exchange, creates that transpiration within the tree, which creates that water draw up and expels the actual gases out through there, and the transpiration and the actual water vapors as well. So, and it's an amazing piece of nature, I guess, the way leaves actually can do that and generate energy for their tree and their crop. So it's really important that the nutrition is correct to create the functionality of the stomata, because if they're not regulating the transpiration and so forth, the potassium's a really key one. There's other elements like copper, phosphorus, sulfur, iron, and nitrogen as well as part of that photosynthetic cycle. But we really need to make sure that they're working well, because if they're working well, a photosynthetic process is working, and we're creating those plant sugars, which are going to drive our system, particularly based around growth and fruit fill. Which leads us into that growth phase of cell expansion. So at this time, we're trying to build canopy, we're trying to build root mats, and overall plant health, which is really also future fruit ripening capacity as well. So we see a slight hormone shift here based around into that auxin growth stage, which is starting to lead over that cytokine and reproductive phase in the tree. So it's about that cell expansion phase. So then we look at varieties of fruit where they sort of are based around that growth stage. So this is where we're starting to see pit lignification or stone hardening just starting to take place. And you can see here on like an early season variety, sort of 34 days after full bloom, where they're starting to sit. And then you can see once they move through to that 40 days, we're starting to get a little bit of early colour change. And then similarly with the late season variety, but just takes a little bit longer to get to that colour change. So this is where we're really starting to rapidly expand cells and starting to load fruit. So if we can have a strong and healthy canopy and strong roots, we're going to get better fruit development through this stage. So some key nutrients to think about through this cell expansion fruit fill stage, things like nitrogen, potassium, iron, manganese, zinc, and boron. So they all have certain roles to play throughout that stage. But once again, we're really trying to maximize cell expansion and create that, really good size fruit and maximize our tree canopy to keep driving that system to maximize our basically elongation of whether it be roots, shoots, or fruit fill. So it's a really short window because that is in that growth phase, which is, like I said earlier, somewhere 40 to 45 days. So with so much going on and so much potential that we need to achieve and such a short window, how do we hit these goals? So, it's a bit like the race car team. How do they stay on track? Well, they start to analyze things. So they'll look at the car, they'll be doing their diagnostics to see how the car's performing on the track. So no different for us. So what we do at Hybrid Ag, we've gone down the path of using a tool for managing photosynthesis, which is called a differential sap analysis. So why we've used this type of tool is when you actually look at how nutrients flow in the in the plant. As you can see on the slide there, we've got a chute which shows basically where nutrients move within those shoots. So mobile nutrients like nitrogen, phosphorus, potassium, and magnesium will see deficiencies sort of appear in the older leaves first, whereas medium, mobile, and immobile nutrients, particularly things like boron, calcium, they're more of the medium ones, sulfur, manganese, copper, iron, zinc, and moly. Well, they'll sort of show their deficiency more in the newer part of the shoot. So when the newer part of the shoot is actually becoming deficient, if it's a mobile nutrient, the plant will actually cannibalize the older leaf and move that into the new shoot to keep it in the best shape it can be if it actually can't get it up by the roots. So when we use our DSA testing method, it's a two-part test. So we actually take a leaf from the newer growth as well as the older growths. So we can just sort of see how the nutrient flows are going and whether there's any imbalances. And what it also does is actually see if you are on track, or if you're not on track, it gives you the ability to actually fine tune and just make some of those adjustments to maximize that photosynthetic capacity of the plant to maximize the crop outcome. And the little guy that comes around the corner, he keeps you on the track, and this is what this is trying to do, is keep us on track. He's looking around the corner to make sure he's not going to run off the road. So then we move into that ripening growth phase. So really, this is the sweetest part of the growth season, arguably the most rewarding, and this is where obviously the fruit starts to soften, sweeten colour, and matures. And this is where all the hard work of the season really begins to start to show that true value. So in that ripening phase, we've got things which are like sugar loading starts to begin, colour change and development, fruit softens, we've got flavour and aroma compounds emerge, we've got some acidic type reduction which is taking place, so we can actually start to eat the fruit when it gets to harvest. So managing the nutrition and water at this stage is really critical to develop a top quality piece of fruit with that optimal firmness and shelf life, I guess, for our customers. And it doesn't matter whether it's domestic or international. It's like we need to make sure that we're doing the right job in field at this time to maximise the consumer experience. So some things that we need in the ripening phase. So once again, when we move into that ripening space, it's a little bit more of a shift back into cytokinin side of things. We've sort of slowed up in all that growth. Shoot growth should have slowed up a bit. We'll still need photosynthesis driving the system. But when we look at our elements that we need or nutrients in that space, it's things like sulfur and manganese and moly, which we actually need as well to actually try and reduce some of that nitrate in the system. And if we can potentially reduce some of that nitrate in the system, if we have rain events, it'll give us a bit more rain tolerance from cracking. Fruit typically cracks when it's really loaded up with nitrate. So if we can reduce that nitrogen in the system, not only does it help cracking, but also helps with sap-sucking type insects and things like that. So we want to make sure that the actual Brix level in the plant or the fruit as well is in a higher concentration, because that's where those sap-sucking insects actually don't appreciate good Brix levels, not only in the sap, but in the fruit. And then we move on to things like iron, manganese, phosphorus, magnesium, and calcium. Once again, it's part of the photosynthesis and part of that cytokinin balance. Things like boron, which are really important for that sugar transport. Cobalt is part of that regulation of ethylene production. Once again, potassium, which is really the hero of the whole thing, is part of that osmotic regulation again. So we've made it to the finish line with our crop. So we might have got the checkered flag, we've got some fruit in the box, and we're all happy. But, the game's still on really, because in the back end of that growth phase, once we've got to harvest, we actually need to be thinking about bud initiation, because this is where that's starting to set up our next year. So bud initiation or floral initiation really happens around this harvest or just after harvest period. It involves that cell division and differentiation within buds, which are forming our next year's crop. So once again, it's important to make sure that we've got key things in the right place from a nutritional point of view that we can maximise his time. So even in this picture here with some fruit on the tree, we can actually see where some of that new developing bud is starting to take place, which will have fruit bud for the next season. So yeah, based around that, calcium is pretty important again, boron, manganese, zinc, and copper, and once again, the biostimulants. So it's a bit of a crossover here of some of those nutrients, and some of them sort of really, like we said, calcium is that one that's in the middle, and he's going to be there for most of that journey based around whether it's reproductive or actually growth. So we'll keep moving into what we call the post-harvest. So it's almost the race team, it's back in the pits, back in pit lane, so it needs to rebuild. Well, that's what we need to do. So when we look at that post-harvest aging phase, so the post-harvest phase is a way that we can start to rebalance. So we look at our post-harvest phase. What we need to achieve here is we've had the yield from the crop and the shoots which have grown from the crop and has taken our storage reserves or nutrients which we've applied. And we need to make sure that the tree becomes back in balanced with, you know, the carbohydrates, nitrogens, phosphoruses, and trace elements to actually do the job from a rebalancing point of view, to make sure they're there, ready to be stored where they need to be in our trunks, bark, buds, and roots for the following season. Because remember, when we go back to sleep or go into hibernation, park the car in the garage, so to speak, we need to make sure the fuel tanks, it's topped up, ready to go. So all those plant processes that take place at post-harvest, and when we get into senescence, we're actually remobilising those nutrients into the spaces that we need to. Because remember, once we get back into that reproductive phase coming out of dormancy, we're relying on those stored reserves to drive that system once again. So it's very cyclic that we make sure we need to top up. So once again, you know, we're really feeding today to fuel for tomorrow. So, you know, nitrogen, calcium, zinc, magnesium, boron, everybody's got a key role. They need to be on the bus, and we need to make sure they're there through that post-harvest phase. Now, when we move into senescence, which is basically that final act, you know, what is different between senescence and post-harvest? Well, effectively, within post-harvest, we've still got leaves which are quite active. They're still photosynthesizing. Whereas when we get into the actual senescence phase, this is where our the age process of actual senescence takes place. So our temperature, day temperature is starting to drop, our daylight hours are starting to drop. We actually have a hormone shift in the tree which triggers that senescence process based around the season. And this creates the leaf degradation, which creates the remobilisation of some of these nutrients which are actually in the leaf, and that's where they move out. We can see our discoloration in the leaf, and over time, obviously, they will drop to the ground and we'll be in that dormancy phase once again. So I guess, once again, this is the back end of the season to remobilise and reboot. So if we missed a little bit on that front end of post-harvest, this still gives us an opportunity here to have another go at it. So things like nitrogen, particularly in forms of urea, which can go into the buds and help for a bit of frost protection that's stored in the buds. Things like magnesium for chlorophyll help that. Also leaf drop, you know, things like zinc. And if we actually do want to drop leaves, we can also add some abscisic acid as well to try and create that, I guess, earlier chill accumulation. Because we know if we've still got active leaves on our cherry tree, we're not really in that dormancy phase until we've really got the sap flow really minimal. So it's really important, and I think there probably needs to be a lot more focus put on post-harvest, because it really does drive that early part of the process of the tree in that reproductive phase until we can actually start photosynthesizing. So once again, once the job's done, then we can park the car in the garage because we've done our rebuilding and the job's done. which is really good. And we can move into dormancy and think about some other things. So just to recap, we've gone through the 4 major phases in the tree, which is quiescence phase / dormancy, our reproductive phase, which is bud and flower, fruit set and cell division, our growth phase, our major growth phase, which is effectively cell expansion, fruit fill, ripening, bud initiation, and then our post-harvest aging phase, which is the rebalancing of our tree post-harvest and drawdown of nutrients, and the senescence phase, which is that last remobilisation of nutrients out of our leaves. So with that being said, if you want to know any more about some of the information that we've talked about tonight, we've got quite a few more in-depth webinars on these various stages. So if you want to grab that link there from the Hybrid Ag Knowledge Centre, it'll take you there, and there's quite a bit of information there that you're more than welcome to have a look at your own leisure. You'll be able to listen to some more of my dulcet tones and some of my colleagues as well. So is there any questions from the audience this evening, or if you want to hold questions back? Jess, what do you want to do?
We might hold questions just till the end, but thank you, Andrew. I mean, I was ferociously writing notes here. I'm in for an education as well. So thank you for sharing and being so open with all the different nutrients that are required. I even put a question in the chat because that role of nitrogen is interesting. But we'll hold it till after Zen. But and if you have any questions, guys, pop them in the chat. That way you won't forget them when we come back to Andrew. But Zen, did you want to try and share your screen? And we'll jump straight in.
Okay. Can you see that?
Perfect. Yes.
Okay. Can you hear me okay?
Yep. Sorry.
You can hear me okay, Jess?
Yeah. Sorry, I went off mute.
That's okay.
That's good.
No, good. No, thank you, Andrew. That was very good. I enjoyed that. So very simple PowerPoint, but very complex topic, which is abiotic stress. So with all plants, there is a universal process or defense mechanism that the plant goes through while it's under stress. And we're talking heat, cold, droughts, waterlogging, UV, anything environmental, weather stresses, that kind of thing. Salinity in the soil counts as one. What happens within plants happens within all plants. So understanding this process really gives you a good overview of how plants handle stress and what they do to mitigate the stress. And as growers, we can learn from that to use certain tools to help the plant get over these kinds of stresses. Interestingly, products on the market favor biotic stress. So we have a lot of fungicides, insecticides to tackle pathogens, to tackle pests. And it's simply because they're a lot easier to make. They're targeting certain things, and we have chemistry that can knock them down. So the market is saturated with products that mitigate biotic stress, and very few that can handle abiotic stress or weather stress. And as we know, weather's very unpredictable and it can be extreme at times, and that is out of our control. But the science now within crop science is leaning very heavily into how plants function in this way and what we can do to help them get over the stresses that they come across. So it is an emerging field. Some of the processes we've known for a very long time, but we haven't been able to create things to stimulate them. So we know, how ethylene works, for example, which is a maturity hormone. We know that it degrades cells. We know that it gives us soft fruit. That's been known for a very long time. But having products that can mitigate the damage of ethylene, for example, we haven't had for as long. So I'll touch on each of the processes that the plant goes through, how it responds to them. So it's very much plant physiology that I'll be talking about, and understanding it will give us a better idea of what products to use that are on the market that can help us mitigate the stresses that we get in the orchards. So a couple of things. One of the first things that happens during stress is we lose calcium. So as Andrew said, calcium is extremely important in overall stages. It is a driving factor for cell wall strength. So very prevalent for post-harvest shelf life, very important for mitigating damage of funguses and pests. Obviously, if we have strong cell walls, these things struggle to penetrate because of the strength of the wall. So having good strength of cell walls is very important across all the growth stages. Unfortunately, when a plant gets stressed, again, universal for all plants, it uses calcium as part of its defense mechanism. So we have here on the cells, on the cell walls, calcium. And there's really only two places where calcium sits within a cell, and within the plant, and one of them is on the cell wall, and one of them is in the intercellular glue that holds the cells together. So if we lose calcium from those areas, we get softening of the cells and sliding of the cells on top of each other, and that's why things go soft and they start to go limp. But the plant uses it. It takes calcium away from the cell wall, it moves it into the center of the cell, and it kickstarts its defense mechanisms. So calcium is extremely important, but we lose it during stress. The second thing that happens is we have a buildup of toxins within the cells of the plant. They're called ROS toxins, reactive oxygen species. Very nasty compounds. There's a number of these, and they get progressively worse over time. So we might start with something like hydrogen peroxide buildup, and it will turn into singlet oxygen. Very, very nasty compounds within the cell. These degrade cells very, very quickly. So the longer we get stress, let's say it's very, very hot days over a period of time, the more toxins build up within the cells. And quite simply, this is what is killing the plant. So how the plant tackles that, we'll get to in a second. I'll go back to this image. And the third thing is ethylene. So the plant starts to produce more ethylene, which is a ripening hormone. And again, we know that it weakens everything. More ethylene, more calcium is lost. There's a direct correlation. And unfortunately, the plant is either upregulating ethylene or downregulating. The switch is either off or on. So when a plant's stressed, the switch is on. We're having toxin buildup, we're having ethylene buildup, and we're actively losing calcium because the plant's using it. So what can a plant do to fix that issue? And antioxidants is one of its defense mechanisms. So we have white blood cells in our system, our biological system. If anything nasty is in our system, those white blood cells tackle whatever it is that's causing us grief. A plant uses antioxidants as a very similar way of tackling issues, particularly ROS toxins. So if a plant has high levels of antioxidants, it can withstand stress for longer, and it can withstand higher levels of stress or more severe stress. And you know, people talk about blueberries are good for you because there's high antioxidants in there, and it's absolutely true. We can't produce our own antioxidants. We eat a plant's antioxidants to get their benefits, and they're part of the plant's defense mechanism. So increasing a plant's antioxidant production, and there are products that can do that, is a great way of reducing plant stress. And again, this is universal for all plants. It can be a blade of grass, or it could be a cherry tree. And this happens within the cells of each. So understanding this process as an agronomist or as a consultant or as a grower who grows multiple crops, it's prevalent for each of those crops. So the same process applies. Now there's a gradient. So obviously the more antioxidants, the longer the plant can withstand stress and the healthier it is, and vice versa. Many herbicides work by hindering this process. So it will either stop a plant from producing antioxidants, so it will naturally die because the toxins build up, or the herbicide is simply the toxins themselves, and we're spraying these toxins onto the plants, which are killing them because there's not enough antioxidants to stop it. So this is a demonstration of the power of antioxidants. The left-hand lettuce had antioxidants boosted, and the right-hand lettuce did not. It was just a control plants. These were put into a freezer over a period of time to simulate extreme cold stress and then taken out again. So the plants that had boosted antioxidant levels withstood the stress of the cold. Obviously still damaged to some degree, but nowhere near as damaged as the control plants, which didn't have boosted antioxidants. So they're very powerful components within plant cells. Different types of antioxidants. There are carotenoids, which are the yellowy orange pigmented antioxidants that we find in carrots. And then there are anthocyanins, which are more of the purple antioxidants. So there are different types, and much like amino acids, plants use them in different levels at different times, and there's a complexity to it on levels. But the concept is what I'm trying to get across, that knowing that a plant utilizes these in this way, if we can use tools that help a plant boost their antioxidant levels, we can help them withstand stress. Now, just going back to this slide here, what happens when we lose calcium? So how can we mitigate that? And the obvious answer is we can apply more calcium, which doesn't really work during times of stress because the plant's stomata are shutting down, it's respiring less, it's taking in less water. Calcium follows water, so it also follows respiration. And even if we did get more calcium into the crop at that stage, it will predominantly get dumped into the leaves and bypass the fruit itself because the fruit is just not respiring as much as the leaves. So just adding calcium or trying to apply calcium at this stage when a plant is stressed, it's not a very effective method because the plant is just not able to absorb it. So we really have to either use a high absorption calcium. There are products out there now which have technology in them which help the absorption of calcium. So they're not just a supply of calcium. There's technology which helps the active absorption of calcium. That's very important. And they can be used at times of stress to help replace what is being lost. And there is also something that the plant does naturally, and it's called polyamines. So if you ever hear polyamines anywhere, they're cell wall strengtheners that the plant produces after stress to strengthen the cell walls. Again, products on the market which contain polyamines. We can use them, we can foliar spray them onto plants. They can be absorbed in and be used in the place of calcium. So they don't function like calcium functions. They are not calcium. So they're not as useful as calcium. But they basically hold the fort for calcium to come back into the position that they were holding, and keeping the cell walls nice and strong. So the plant's producing antioxidants to mitigate the toxins. The plant's producing polyamines to keep the cell walls strong. And it's also, after the stress has gone, down-regulating ethylene. So all of these nasty compounds that are being produced during stress, they have to be mitigated after the stress is gone. The plant has to turn them into less damaging compounds. And that takes time. It takes resources, energy for the plant to do. And that's why often after a stress event, it might be a drought period, it might be, periods of prolonged rain and overcast weather. The plant doesn't just recover instantly after the stress event. It takes time. It's working at reducing these toxins and turning them into things that aren't as damaging. Now, we can help facilitate that to some degree by making sure that the plant has the right nutrition. So following Andrew's examples there of having the right nutrition at the right stages gives the plant the ammunition it needs to rebuild after a stress, to recover after a stress. Very much like us, if we're ill, if we don't have the right nutrition, if we don't have the right vitamins and minerals, our recovery is a lot slower, especially if our white blood cell count is low, our immune system is low. It will take us a lot longer to recover after stress or after we've got ill. And it's the same for a plant. So we do need to make sure that we give the plant everything it needs to recover. We need to make sure that ethylene is not on. So there are quite sophisticated things out there now which can turn off ethylene, just like we have products, Ethafron, I believe, is one, and a few others that turns on ethylene, or spray is an ethylene. So we can spray a plant with an ethylene product. We get very good coloration and very good Brix levels because it's maturing from the ethylene. But the downside of ethylene is we get softness or lack of calcium. It degrades the calcium in the cells. So we need to, if we can, reverse that, turn off ethylene and help the plant recover quicker. So I will get to... so this is just a point on antioxidants. So anthocyanin antioxidants are also responsible for coloration in the fruit. And this is an example from China where they boosted the top image with antioxidants, anthocyanin antioxidants, and they didn't for the bottom image. And when there was a high stress, very, very hot days, the bottom plants, the bottom fruit metabolized the antioxidants within the fruit. So it was using these antioxidants to defend itself, and in turn reversed the color or stopped the cherries from ripening as well as the antioxidant boosted cherries. So we see this in apples, and we see this in mangoes, and we see this in a great many crops that have a color change. When there's a stress event, they struggle to get that color and the reason is that the plant is utilizing a lot of those pigments, those antioxidant pigments, for stress mitigation. So again, very, very important. Now, soft cell walls, that leads to a whole wealth of issues. So you can have all the adequate nutrition in place, but if the crop is stressed, it's not utilizing what it has available. It's not taking in as much water. It's not taking in as much nutrients. It's not metabolizing the nutrients it does have. It's actively losing calcium. So we get softening still. We get cell walls weakening, more bacterial infections, more fungal infections. Andrew touched on it with sap-sucking insects that have a proboscis and skewer the cells. We get more pest damage, and we get more cracking because the cells are weaker and they can't handle any expansion or any stresses, so we get more cracking. So having very strong skin or even cell walls within the leaf structure mitigates a great deal of issues, disease issues, pathogen issues. So helping the plant overcome stress isn't just so it doesn't slow down, and it also has a knock-on effect for a huge amount of these nutritional elements too, and pathogen elements, and again post-harvest. So the last thing we want is to have poor nutritional values in the fruit when it's being picked, because it's not going to last very long. It's going to get soft and bruise easily, and get funguses and things like that. So keeping the plant healthy has a knock-on effect, quite a significant knock-on effect. And to give you an idea of just how much, 80% plus of global yield loss is due to abiotic stress. So not biotic stress and not deficiencies in nutrition, but simply weather or environmental stress causes the majority of yield loss, whether that's flower abortions or fruit abortion, or whether it's the plant growing very slowly, or the roots not functioning properly because they start to shut off during stress, or it could be more susceptible to diseases and deficiencies because of the weakening of the cells. So it's very, very important to understand the plant's natural processes, try and find things that help facilitate those processes, and allow the plant to come through those stress events as unscathed as possible. So this is a quite extreme example from University of Lancashire, and different crop, different plants, but it gives an example of how powerful anti-stress products can be. So this is a bean plant that's been herbicide sprayed with paraquat, which is quite a nasty herbicide. On the left-hand side, it was watered and then herbicide sprayed. On the right-hand side, a stress relief product was used, and then herbicide sprayed. So a little bit of damage on the right-hand side where the product has been applied, but survived and kept the plant healthy even after a very severe herbicide has been used. Example again, I don't know if you can see that. Is that coming up on the screen there? Yep. So tomato plants, just as a laboratory experiment. Top left, just herbicide. Top right, herbicide plus an abiotic stress reliever. Bottom left, control plant with nothing. Bottom right with just the stress reliever. So again, still some damage from the herbicide on the top right, but survived. The plant came through, could still produce, got through the stress event. This is a very extreme example of stress, probably the most extreme that we can force on a plant in a laboratory condition to demonstrate this. The second would probably be a very severe salt stress. We would probably add a salt water to the irrigation to stimulate salt stress or drought stress and demonstrate the same thing, which has been done many times. So technology is changing. We're understanding how plants defend themselves better. Thankfully, the process is universal, so it applies to all plants, which means stress relief products that are coming onto the market or on the market now work universally on many different crops. They're very efficient. The older technology, which still has a part to play, and they're things like seaweeds and other biostimulants which help a plant through stress. They've been well known for decades, and they really function because seaweed grows in a very stressful environment. It grows in salt water. So for a plant to grow in salt water, it has to have very robust defense mechanisms to keep it alive. And we can manipulate that. We can use that in products, agricultural products, to use on other plants. We can borrow that biological technology, let's say, and apply it to other plants to help defend themselves against stress. And they work. They're very well known for helping plants stay, rigidity in the cell structure, and they have many modes of action. But technology is changing, and we're having more and more advanced tech, which is coming out in different fertilizers and different biostimulants, which can very much target what I've been talking about on the cellular level. So considerations for when to apply these things is really down to the growing conditions. So around flowering is an optimum time to use these things because they're a critical time for the plants. They dictate a great deal, like Andrew said, about the future of the yield, and getting it right at that stage gets it right at the end. So if we have severe stress hitting the plants during flowering, for example, it has a huge knock-on effect of how much of those flowers will turn into viable fruit and how well developed that fruit is going to be. And also timings as well. It might prolong all of that too long and we start getting a lot of issues. So around flowering, up to pea-sized fruit is absolutely critical for mitigating stress. And I would say at the end of, close to the end of harvest too, because that has a big impact on quality. We don't want cracking, we don't want soft fruit, we don't want discolored fruit or not adequately ripe fruit. And we want very good shelf life. We want a nice, firm cherry. So they're two critical timings. I would say that you should be applying some abiotic stress relief during that time as good practice. And there's always some stress happening within the crop. So whether it's at the root zone level, maybe salinity in the soil or high ECs, or it could be environmental. We could go from a very hot day to a very overcast and rainy day, which is a stress event, or we could get high UV levels, which we're prone to here in Australia, and a whole wealth of other conditions, waterlogging and whatnot. So there's always something going on where the plant's having to deal with, and for us to not just get the nutrition right, but to help the plant through that hard time so it comes out of it without knocking onto the yield. Jess, how are we doing for time? Okay?
Probably got about 1 minute, Zen.
Okay, I saw you lean forward a little bit, which is usually my cue. So the takeaway message really is, I'll go back to this slide here, which just encompasses a bit of everything. Just understanding the process of what the plant's dealing with. It's a living thing. It has a living system. This is part of its defense mechanism. We're really understanding that now and helping the plant get through it. So getting the nutrition right is 101. They're the building blocks. That's the ammunition the plant's using to build good fruit and then that nutrition as well has a massive knock-on effect with hormone levels, which dictate where the plant grows. And that's a whole different topic, which we could probably talk for about 3 hours between us all about that. But the takeaway message is if we can knock out these issues that the plant's having to deal with, toxins by having high antioxidant levels, lack of calcium by either using a good calcium absorber or a polyamine product to help strengthen cell walls while the plant's struggling, and then turning off ethylene so the plant's not in downward spiral. It's actually coming back to recovery mode. So it's an extra consideration for people on top of nutrition.
Awesome. Thank you so much, Sen. I hope everyone got something out of that. We went a bit deep into the physiology and the actual antioxidants response, which, really interesting for myself. So I hope everyone else found it valuable. We've just ticked over 1 minute past 8. Now I know Origin starts at 10 past 8. So if everyone holds on for 5 minutes, we'll run through the quick questions, but I'll get our two speakers just to be quick with their answers. So we'll start from down the bottom. We've got, if we start using antioxidants like Indra on fruit, is it approved for international markets? So can we use that with MRL with holding periods? And how useful is it for frost damage prevention or mitigation?
Yeah, good question. So, yes, it's approved for international markets. It's used all around the world. That's a product from Levity Crop Science, which is a UK-based foliar fertilizer company. Very powerful abiotic stress relief product. We use here in Australia. It's used in South Africa for cherries, and the United States for cherries, and also in China. So they've probably been using that internationally for about going on maybe 10 years now. So we're a bit late to the party in Australia, but it works very well internationally for abiotic stress. What was the second part of that question? So does it work for frost damage?
Yeah.
So no. Frost is very much a physical damage as much as anything. Antioxidants and different abiotic stress relievers won't help with physical damage, which is more of a biotic stress. You'll still get punctured cells from frost spears and the physical damage of the frost on the cells. But it will help with the internal recovery of the plant after the frost has gone. So you will still get damage with the frost, but it will help with the recovery thereafter. So we use it just after the frost has been, or we use it beforehand if we can predict the temperatures, and then it helps with recovery. So with frost physical damage, it's more things like silica, which is a physical strengthener. We get some good results with silica, for example, with strengthening things physically. And again, calcium levels, potassium is used to help that freezing point. And there's a couple of tricks of the trade that you can use to mitigate the actual physical damage of the frost. Seaweeds are known to do that to some degree as well, but this is for recovery.
Thanks, Dan. I think as well, Karina, your question about critical time to apply, you're happy with the answer of around flowering. Yep, you're happy with that one. Cool.
There's a point there, Jess. Do they require continual application? So plants, thankfully, can store antioxidants. So there are a few elements like potassium and phosphorus, which plants can store as well in the vacuole of the cell. They can store them for a rainy day. So if we overdose with these elements, including antioxidant products, it can be stored. So we can apply these things up to a month in advance of the stress event. So 3 to 4 weeks in advance in many cases, which is fantastic because it means we don't have to get the timing crucial. We can apply it roughly before, okay, in a few weeks we're expecting very bad weather, or there's some kind of weather event coming in, and we can kind of predict it. So.
Awesome. One for Andrew, forms of calcium used and foliar versus soil applied for best effect.
Yeah, that's a good question. I think both in unison work well together. I think you've got to pick what you're trying to achieve based around what's your soil profile. When I say what you're trying to achieve is like, do I have a soil that's actually going to work if I put calcium into it? Meaning, is my soil balance right, or do I have salt, or do I have high mag, or to go understand the synergies or the antagonism that could be going on in the soil. But if you've got everything in balance in the soil is a good start because, as we know, it goes up in the water pathway. But foliar is a good secondary, particularly early on. So they both have their place.
Great. And would you do a leaf test at ripening phase?
A leaf test?
During ripening phase.
Yeah, absolutely. If you're at harvest, it's a scorecard to see where you got to in the journey. It's the ultimate. It's like you cross the finish line. Well, how did we go? It really tells us where we did finish in the race. We might have thought we come first, but maybe we come 5th or 6th.
And last one for Zen, you talked about seaweeds and biostimulants being old technology. What are some of the new products that could be used?
So old in the sense that we've used them for a long time. Plants have been using, obviously these elements for millennia, and they're fantastic. They're from the plants themselves. So, they're high tech in the sense of biological systems. Old technology in the fact that we've had them in agriculture for a very long time. Now, there are some products on the market which target the stresses on this cellular level, which I'm talking about, and are less of a generalistic, like a seaweed, for example. So I believe Stoller has one called Bioforge. That was a precursor to Indra. Indra is probably the latest that from Levity Crop Science, which is a litre a hectare monthly. So you don't get much more efficient than that when it comes to a foliar product. Now, because stress, it's linked to so many things. So if you get the stress product on, but the nutrition is wrong, the plant doesn't have what it needs to come out of that stress adequately. And again, from a root level perspective too, if your irrigation is not right, or your composition of nutrients within the irrigation is not right, it's too much of this or too little of that, the plants again can't utilize what they have properly. So we have to get as many of these elements correct as we can. Irrigation is a big one. Irrigation itself can be a stress event if we have prolonged periods of drought or poor irrigation, or vice versa. If we're over irrigating as well, that's a stress event for the root system. So we can apply very good products, but we're counteracting them with poor irrigation practices or poor nutrition.
Thanks, Zen. And I guess if you have any other comments, we can pop them in the chat, and Zen might be able to get to them. But we'll call it stumps here. A massive big thank you to Andrew and Zen. They took their time out of this evening, tonight, and also prepped those presentations for us. So really appreciative of them, and I hope you guys got something out of this session.